All right, guys, welcome to our first screencast full lesson. So this is for 9.1, Transport in the Xylem. This is an HL only lesson. So if anyone in SL is watching this, you can turn it off and don't worry about it. So we're going to be talking about the transport in the xylem. So if you look at our list here, we need to talk about the fact that transpiration is a consequence of gas exchange, that plants transporting water from the roots to the leaves, they have to replace the loss of water from transpiration. They do that by transporting water they get at the roots. We can talk about the cohesive property of water. That's back to topic 2. And the structure of xylem vessels allows that to happen. We can talk about adhesive property of water, which is going to make the tension forces in cell walls, which is an important part of how water is transported around plants. We're going to talk about how different imaging software has allowed us to better understand how this works. Talk about how minerals are taken in by the roots, and then how water absorption by osmosis at the roots. It's all about water and minerals moving around plants today. Applications, we'll talk about plants who live in deserts and saline soils and how they deal with that for water conservation. We'll look at different models, or we would, if we were in class, we would do models using blotting paper or filter paper, that kind of stuff to model some of these properties. But of course, we can't do that virtually, so we will do it later on. You're going to draw xylem vessels based on microscope images. And the last two, the skills here that you see with transpiration in Protomus Practical 7, Okay, these two here, and experiment test hypotheses. This one you're going to do when we come as a virtual lab, and then we'll do again in person when you come back. And then the bottom one, the skill, that's your kind of homework for today to submit on Schoology, is your experiment you have designed individually to do this. Okay, so let's talk about the structure of leaves. So if you take a look at this leaf and you've done your reading, what are the key functions of a leaf? Pause the video and think about it. Okay, so you thought about it, maybe talk to somebody through WhatsApp or something. So key functions of a leaf be the fact that we are going to have photosynthesis happening. That's the main one, that in these cells here, photosynthesis is going to happen in chloroplasts. And that's the number one thing they're going to do. We also have gas exchange happening, where we have to have gases coming in and out of the stoma to allow that to happen. And then the sugars produced in the leaf during photosynthesis, they need to leave through what's the vascular tissue here. The xylem and phloem will take those sugars where they're produced to where they're needed in the plant. Okay, where and why does transpiration happen? Again, pause the video, talk to somebody, or think about it yourself. Okay, so transpiration is happening in the guard cells. So in the stoma, which are open and closed by guard cells, the bottom of a leaf. Most leaves only have the stoma on the bottom. That's to avoid excess water loss. And it happens because we have to have an opening for gas exchange to occur. We have to have a spot for carbon dioxide to come in and for oxygen to come out, right? So I have my CO2 going in, and I have my O2 coming out. And unfortunately, because those guard cells have to be open for that to happen, I'm also going to lose a little bit of H2O as vapor. And it's an unavoidable consequence of respiration, of having the guard cells open. Okay, now if we look at the structure of a leaf, how does this structure of a leaf support its function? Well, let's just get rid of this colors here. So the function of a leaf is mainly photosynthesis, right? And all the things that require photosynthesis. So if we look at the picture here, the part of a leaf that is biggest is we have what's called the blade, right? The blade of the leaf is this big part of the leaf here. And we have these structures in the leaf, think of them as called the veins, which hold the leaf open to allow a lot of sunlight. It's a high surface area and it's very thin. And the goal is to have all of these cells at the top, which allow photosynthesis to happen. Notice how it's all these tightly packed cells at the top for photosynthesis, and then we have these more spaces here. So the top ones, these are called palisade mesophyll cells, And that's where you're going to get a lot of photosynthesis happening. It's close to the top, closer to the sun, all that's going to happen. And then below, we still have some mesophyll cells, but there's more spaces between them, and that's a spongy mesophyll. And that's because the gases produced in photosynthesis have to have a space to get out and then out through a stomata, or have to have space to get in and where it needs to go. The gases we just talked about. Also part of the structure is the vascular bundle here, several of them in the leaf, and that's going to bring materials like water from the roots up to the leaf so we can use it for photosynthesis, and it's also going to bring sugars and the products out. So it's going to bring sugar out, or energy, we can call it energy. Okay, next question. Is the structure of a leaf the same in every type of plant? Why or why not? Take a minute and think about that. Okay, so some plants have very large leaves with really large surface areas. So that way they can collect lots of sunlight. Some plants have very small leaves. For example, the spines on a cactus, those are actually modified leaves. So a major factor that affects this is the environment, right? Plants that are in an environment where there's lots of water and they're not going to worry so much about water loss, so they're going to have bigger leaves with more stomata. Plants that have space and don't need to compete too much are going to have bigger leaves. There's all sorts of different adaptations here. We'll talk about more specific ones in a little bit. So that's the end of our kind of leaf structure lesson. So take a minute, go through your notes. We've been very thorough. If you want to pause and go back the video and listen to it again, you're welcome to do so. We're going to kind of test your knowledge now. So here is a diagram of a leaf with some labeled parts, a little bit simpler than what I just showed you. It's black and white. And I want you to use this to talk about adaptation of function. So structure and function is a huge theme in this entire course. The connection between structure and function. Let's try again. There we go. Okay. So here's a list of the different functions of a leaf: to absorb light, photosynthesis, gas exchange, conserve water, transport products out. We talked about this a little bit together already, but I want you to try and do this by yourself. So an example of a sentence you can say here is the waxy cuticle, it's right down here. Example, the waxy cuticle of a leaf helps water conservation. It prevents water loss in the upper epidermis. So take a minute and write me some more sentences about how the functions of a leaf are met by its structure, illustrated here. Pause the video, give me some answers, please. Okay, the second analyzing a leaf task here is here we have the same kind of leaf diagram, but it's highlighted to focus on the movement of water. Okay, so how water moves through the leaf. I need a stylus, there we go. Okay, so you notice here it's talking about how the xylem is going to bring water, that's a part of the xylem right here. How when that water arrives, here's a little arrow showing its movement. It's going to move through the cells to the very thicker cells up here, the palisade mesophyll, through the spongy mesophyll, out here towards these kind of open spaces. It's going to be absorbed by cells along the way. That water will then evaporate from the surface because of heat and will be let out if the guard cells are open of a stomata. If the guard cells are closed, it's not going to be let open. So use that information and answer these questions. Pause the video, answer the questions. Okay, so here are some sample answers to the first question. So you notice here there's highlights showing each function with the structures in the same color on here. Okay, so parallel ones, we have blue and yellow, and some sample answers here for the palisade mesophyll are they absorb light by photosynthesis, making glucose. That spongy mesophyll cells absorb light by photosynthesis, and they also carbon dioxide from the leaf airspace. That water conservation is promoted by closure of stomata by guard cells in the lower epidermis. Remember, the epidermis is this bottom part of the plant, the kind of almost like the membrane, if you will, the outer layer of the cells, of the tissue. So check your work. And check your work for these questions here. Sample answers. Okay, so that's leaves. Let's talk about minerals and water from the roots. So as we're losing our water through transpiration and we're using it in photosynthesis, we have to replace it. And where do plants get their water? From their roots. So how that works is it's through the xylem that the water will move. So if you look at this vascular bundle here, you have the blue X, that's the xylem, okay? As we're focusing on today, we'll talk about phloem and cambium later on in this unit. So it's going to absorb not only water, but also minerals through the roots, through the xylem, and bring it up the plant. Okay, it's also very important in supporting the plant. So the vascular system includes xylem, phloem, cambium, this whole central part here, and non-vascular plants. You remember from topic 5, the non-vascular plants are the bryophytes or the mosses. They cannot grow very big. They can only be a few centimeters in size because they don't have this xylem and phloem to help support them. Okay, now take a look at this diagram, and we have here a root kind of zoomed out with the root tip with the root cap on it. And then we have little root hairs here, and this is a zoom in of that part. So here's the root hairs on that. So pause the video to answer this question. What are the functions of the root hairs and the root cap? Answer this question here. Okay, so our functions of the root hairs would be to significantly increase the surface area of the root, up to 3 times larger surface area, and more surface area, as we know from talking about human physiology, among other topics. More surface area means more absorption. So it can absorb more water and more minerals by having more surface area. The root cap is going to protect the root tissue as it burrows down into the soil. So roots are constantly trying to go down further in the soil, either horizontally or vertically, to try and reach more areas to get more water and more minerals. And that can damage the delicate plant tissue. So root caps help protect the root. Okay, let's talk about xylem's structure. So there's 2 main components of xylem. There's a vessel elements and there's tracheids. Okay, so there's 2 parts here you need to think about. So the vessel element and the tracheid. Okay, tracheids are 100% dead cells. There is nothing in them here. Okay, so here's your tracheids up here. These ones are your tracheids. And they have these tapered kind of ends that fit together to make a continuous column. And they're really important for support and structure of the plant as well as for movement of water and minerals. Vessel elements are also dead cells. Okay, so xylem is dead. And they have really thick, very major walls. They have primary and secondary walls. So if you look here, vessel element, you can see this extra wall here. Okay, and their walls, especially what they have pits. So if you look at this one here, you can see the pitted walls, little kind of holes, and that allows for lateral or horizontal water movement. Now water is going to move up the plant through these different components of a xylem. If you look here, here's a vessel element, and here's the tracheids around it. Through this whole process, through a method that we describe as called the cohesion tension theory. That explains how water can move up a plant. It's not being pulled using energy, it has a whole bunch of properties of water and the plant structures that allow this to happen. Okay, so before we talk in detail about cohesion tension theory, what I want you guys to do, one of the skills from today is to draw xylem from a microscope image. So if we were in class, we would actually look at this with a microscope and some prepared slides and look at yourselves. But unfortunately, we can't be in class, so instead you're going to be looking at these images here, which are from microscopes, and you're going to draw then into the diagram. So you're going to draw it, edit it, and upload your drawing to Schoology. So I want this one's Schoology as well as your kind of lab design, both uploaded to me, please. So take a picture of it once you draw it and upload it. Okay, key ideas. So our key idea so far is the structure of a plant's leaves, roots, and vascular system called the xylem allows it to move water and minerals around the plant. And the molecular properties of water are an integral part of the movement through the xylem. So let's talk about cohesion tension theory. So in topic 2, we learned about the properties of water. And those properties are really key to how water can move up a plant. So how do they do that? Take a minute and think back to topic 2, think to your reading, and try to answer this question. Pause the video. So one way in which they do that is through cohesion. And remember, cohesion is the hydrogen bonds between water molecules. So if we look at this diagram, it's these little bonds here. That was really poorly drawn. These little bonds between the water molecules. Okay, those are the hydrogen bonds that make cohesion, and it helps keep the water in a continuous column of molecules all the way up the plant. The other one is adhesion, and that's what you're going to see over here is the water molecules connecting to the wall of the plant, the vessel side, the xylem. And that adhesion is what defies gravity, allows the water molecules to not fall down the plant when they're not being pulled upwards. All this movement of water is done to maintain a concentration gradient. So we're trying to keep the concentration of water equal throughout, and so as water is lost through the leaves up here, through the stomata, it has to get pulled up from the roots to maintain that. Okay? So how this kind of works, let's talk a little bit more how this works. So I have water being absorbed at the roots and taken in, and those water molecules will form a continuous chain using their cohesion, okay, to each other. And then they will also hold onto the sides of the xylem through adhesion. And then as the water, that continuous chain of water, if we kind of show it here in the molecule, as water is being pulled out from transpiration at the leaf, that will just keep pulling the chain upwards. Okay? So it's going to continuously move the water upwards across. The xylem is going to also, the tension part of this model is the fact that as we have this tension of the leaves are, let's articulate this. This tension from transpiration, this like pulling of the water, is maintained throughout the entire plant because of the solid structure of the xylem vessels. Because they are so rigid, it allows it to maintain that tension. So we have the cohesion and tension being able to pull the water all the way up the plant. Okay, let's talk about the stomata and the guard cells. Very important part of how water movement is regulated in plants. So first of all, what has to come in and out of the stoma? Take a minute, pause, and think about that answer. Okay, so hopefully you got the answer. What has to come in is carbon dioxide. What has to go out is oxygen when the plant is photosynthesizing. And then water loss is a kind of an inevitable part of that process as well. Now don't forget, plants also do respiration, but they tend to do it mostly at night. And they're going to do the opposite. They're going to use carbon dioxide and use oxygen and produce carbon dioxide. Now, if you take a look at the structure of the guard cells here, notice how this part, the interior part of the guard cells, that's much more thick. It's much thicker towards the stoma in the center than it is in the outside. And it's because that's the part that is going to kind of become flaccid and kind of fall in to close the opening. So when these are full of water, notice in this one is lots of water molecules here, and so there's not as much water. So it's full of water, it becomes quite rigid and it kind of pops open. When the water leaves, and there's little red dots are the water, when the water leaves, okay, then they become much more flaccid, they don't have enough to support them, and they kind of close shut like this. Okay, so water is what helps keep them open and closed. Now, what causes the water to come in and out? Well, potassium. Potassium ions are the key in this factor, and what's going to happen is when blue light, the blue part of the visible light spectrum, when it hits the stoma, it's going to trigger active transport of potassium. So we're going to spend ATP, we're using active transport to bring potassium into the guard cells. Okay, so potassium out here, so potassium's red, is being brought in through active transport because of blue light. And with a higher concentration of potassium, that's going to change the concentration gradient of potassium. So then water wants to fix that, wants to make sure everything is equal concentration. So water is going to move in by osmosis to try and reach equilibrium. So by actively transporting potassium, we can use passive osmosis to move the water in. Okay, and when potassium leaves the cells, so does the water. So potassium will naturally try to go back out again to be able to maintain that concentration gradient. And when it does that, water also leaves. Okay, now there's a hormone that causes potassium to leave the guard cells very quickly. Okay, so when potassium leaves the guard cells, what's going to happen to the opening of the stoma? And when white plants make this hormone? So pause the video and think about this. So if the potassium leaves quickly, then the water is also going to leave. And when the water leaves, the whole stoma is going to close because now it's become flaccid. There's not enough structure there to hold it open, so it just kind of closes on itself. And that means you're going to close the stoma, we're going to reduce water loss. So this hormone, abscisic acid or ABA, often it is produced by the roots of a plant. And it sends the hormone signal because the roots aren't being successful at getting enough water to keep the plant hydrated or what to meet its needs. So it sends that signal all the way up the leaves and it causes stoma to close, which is really cool how plants that can be very, very large are able to communicate from the roots to the leaves based on water, what they're finding for water. Okay, so here's the key idea about this. Stomata open and close to control transpiration rates based on environmental cues. So the next kind of thing we're talking about here is how do we know? How is this going to happen? Well, the technology we have and have developed over time has really helped cement our understanding of how this happens in plants. So take a look at these three kind of images. We have just a photo that's zoomed in. Oops. Okay, a zoomed in photo. Then we have a compound light microscope. You can see this in class, this kind of level with our microscopes we have in class. And then an electron microscope, much more detail. So how does the level of understanding change as we improve the technology of our images? Take a minute, pause the video, and think about it. Okay, so as we can see, we couldn't even see, before there were microscopes, we couldn't even see that stoma existed. So there's no way we could do that. And then as technology improved, we're able to kind of see the details of the structure, the opening, how they're going to swell open and close. This one's, if you notice, it's kind of into a sunken pit. That's a strategy to reduce water loss. So more projected on top. The difference, all those differences we can find only with better quality images. So if we don't have a technology to see these things, we can't understand them. Now, processing software, if we are able to then take these images and use digital tools to measure them very precisely, the size of the opening of this one compared to that one, maybe the thickness here, if we can measure those things with an electron, with digital image software, that's much more detailed and specific and accurate than we can do by hand. There is a lab I'd like to do where you guys actually remove the stoma from leaves, and we will do it, but can't be done virtually. We'll do it once you return to campus. Okay, on our last things for today, this long lesson is moving minerals. So it's not just water being moved through the xylem, it's also minerals. So when would minerals move passively? What's going to cause a passive transport of minerals from the soil into the roots? So from the soil outside here, when are they going to move from here into the roots? What's going to cause that to happen? Well, it has to be that we have a concentration gradient, right? It's diffusion, it's a passive transport. There has to be a higher concentration of minerals in the soil than there is inside the root, inside the plant. It can also move even if the concentration is not correct, but if the water is moving in like a bulk flow or a mass flow of water, large amount of water at once, it will often take minerals with it. And that can also cause a passive transport of minerals. Okay, if that's when they're going to move passively, when would minerals move actively? Well, we know active transport happens when we're moving against a concentration gradient. So if I have lots of minerals inside the root already and low concentration in the soil, I'm going to have to spend ATP to bring those minerals in. Another reason is if those minerals, for whatever reason, their polarity, their size, they can't fit through the membrane, then they have to go through special cells, membrane proteins that will allow the minerals to come into the cell, and that also takes energy. Potassium is an example of a mineral absorbed that way. Okay, so let's talk about how these different plants adapt to their environment based on these different features. So, every species on this planet has evolved to their specific environment. We talk about a lot in topic 5, and in this one, I want you to talk about the adaptations to dry conditions, low water conditions, and to salty conditions. So, succulents and cacti, those are two types of plants, xerophytes is the name of the family, that can store water and prevent water loss. So here's some examples of structures here. Some cacti have these really huge bulbs underground to help them store water. On a cacti like this, the spines are actually used to be leaves, or modified leaves, but they have no real leaves, so they don't want to lose water through transpiration. Here we have the prickles and also help prevent animals from biting the plant and taking the water that's stored inside this big fleshy part of the plant. When it rains in the desert, if you haven't experienced it before, it rains a lot for a very short period of time. So it's really important that xerophytes are able to absorb that water right away. So a lot of these plants have this kind of surface layer of root that goes very horizontal and not very deep, some of them, because it's going to allow them, when it does rain a lot, they can absorb all the water over this whole area very quickly instead of just going directly below them. Aloe is a single plant that's of xerophyte, and it has these fleshy leaves that it stores water in, and that has a waxy layer on top of it called the cuticle, and that also helps prevent water loss because the water can't move through wax. Like water is of course polar, and wax is nonpolar. In some places, these plants will have roots that go very, very deep down, so they can reach the waters in the water tables, deep down underneath the earth, and what's called the aquifer. It can be stored there for a long time. So those are some adaptations for water loss. Another kind of plant that has to live in a certain environment are mangroves. So mangroves live in what's a salty water environment. It's not the ocean, but it's a kind of ecotone, a bridging area between the ocean and a river or a lake. And so the water there is a little bit salty. It's not as salty as the ocean, but not fresh water either. So one thing that these plants can do, that mangroves can do, is in their leaves they have special pores. They can actually absorb that salty water, and they can remove the salt, so they can get the fresh water they need to survive. Some plants can also separate the sodium ions in the salt, separate it away from the chlorine ions, so it keeps them from actually being together. So if we separate them inside of a cell, it can't make salt, and that's one way of protecting the plant. Okay, key idea, the last one, is plants have evolved structures to access and retain water in several different environments. Okay, and this is your final task for today, is you need to write for me a methodology, outline methodology, how you're going to test how either temperature or humidity is going to affect transpiration of plants. So I want to see methodology where you have what's your procedure going to be, what are your constants, how you're going to collect your data. Here are some examples of how you can collect data. For this kind of experiment, you can just put a bag on a leaf and see how much water is produced, comes off from evaporation and then transpiration, and then condenses into water. You can do a change in mass. You can count the droplets of water. There's a ton of different options you can do to think about what you want to do. How you're going to collect data, and how you do this. So on to Schoology. For today's lesson, what you need to give me is you need to give me this methodology outlined, okay, as well as an upload of your drawing of the xylem of the microscope image. Okay, so that's your task. for this lesson for this week, and then after the March break I'll be coming back and you'll be doing 9.2. So your homework for the week after next when we come back will be to read 9.2, Transport and the Phloem of Plants. All right, guys, hopefully that was helpful for you and you get some good information and you're able to take good notes on this.